Alright, so in this video I want to talk a little bit about mitosis, but before I jump into what mitosis is, I want to give you some context as to why we're going to learn about it. And you might already know something about mitosis, and you might already think that you know something about mitosis. Either way, it's going to be really important that you pay attention because we might be actually breaking down some misconceptions that you might have about what mitosis is. But I really want you to understand that this whole thing is really all about cell growth, right? So this is all about growth. And if cells are able to grow faster, what they often do is actually divide and make more cells. And so when we think about an organism that's really big, like you, me, whale, a plant, we can think of them being made out of many, many cells. It's not one large cell. Plants will actually, all living things, once they get to a certain size, will make their cells divide. Of course, they're using some of their DNA that they've inherited in order to control for that action, but they divide, and our explanation for that is really about surface area and volume ratios. We want to maximize the surface area, reduce the volume, and that ratio is critically important so that we can get things in and out of cells quickly, and they don't have a great distance to travel. And what necessitates a high surface area to volume ratio is actually a really small cell. Or some cells have some adaptations that we can talk about if you bring that up in class. So make sure you ask some questions about that. When we think about this growth, a way to think about this as an organism gets bigger, that growth could be looked at or viewed as cellular division, right? Or cell division. And there are several ways that cells divide. It depends on what type of cell we're talking about. So if we're talking about, let's go back to some words that we've learned about earlier this year, eukaryotic cells and prokaryotic cells. Eukaryotic cells can divide in a multitude of ways, but the two major ways that we will end up talking about is through mitosis, which is what this lesson is really all about, and the other, depending on the type of cell and what the function of that cell is, would be meiosis. Whereas prokaryotic cells largely are going to be dividing through one single process, which is called binary fission. And this is really, again, like level one understanding. We're going to then add some complexity to that in class and as we continue the conversation in our program. So what we're really focusing on is a type of cell division that's called mitosis. But I have to admit that I've been lying to you a little bit. That mitosis is not really cell division. It's really about nuclear division. And that's critically important. So what we're going to do is go ahead and dive into this, but I think the point that I want you to understand before we jump in is that we could actually look at cells under a microscope and figure out how quickly they're dividing by counting the number of cells that are in mitosis versus those that are not in mitosis. And we can think about proportions and relationships of types of cells, and we'll get into all of that. But let's talk about what the context of mitosis is and how you might have been misled with your idea that mitosis is about cell division. All right, so I'm going to take that off and add a new layer here. And I want you to draw a circle in your notes, right? So I want you to draw a circle. Make that a circle as best as possible. Got my center point here, and by no means is this the scale. I'm not doing it to proportion. I'm just drawing this so that we can include all the information, and we'll talk about what normally happens proportionally. So I'm drawing a line straight up and down so that I've got 3/4 of the circle in one section of, in one lined off or sectioned off portion, and then I've got a 1/4 up here. And I'm going to do these dotted lines across so that I have my 4/4 here. And what we're going to do is add some arrows so that we're talking about this cycle in a clockwise manner, right? It's going clockwise. Of course, we could represent this in a counterclockwise method or some other diagram altogether. And what we're going to do is call this the cell cycle. And I want you to be wary of that. So anytime I say I'm saying that this is the only cell cycle, and that is not true, right? So we're going to use this as our basis of the cell cycle. And then, again, add complexity. So what we're going to do here is label this whole section, these 3/4, as interphase, and there are 3 sections to interphase that you might already know, maybe, G1, S, and G2. And that's critically important for us to understand because it's during these 3 phases that the cell is doing its normal job. It's not worrying about division. It's not worrying about reproduction. It's not worrying about anything other than, do I have the reactants that I need for all my reactions? Am I getting them quickly enough? Am I getting rid of the products of the reactions? And am I functioning, right? Like that's what the cell is worried about. And again, this isn't going on in each cell in your body. This is kind of like the timeline or the lifeline, the life cycle of your cell. So this G1, just to review a little bit about what's happening in G1. So in G1, we would actually have started with a small cell, and by the end of it, we end up with a bigger cell. So the cell grows in size, right? So that's one thing that's happening. But the second thing is that the cell is also increasing the number of organelles. So as the cell gets bigger, it also needs more parts to do more jobs. For example, it needs more endoplasmic reticulum. It might need more mitochondria. If it's a leaf cell in a plant, it might need more chloroplasts. And this is when that's happening. Not where, when. And then the third thing that's happening, which is critically important, that often derives the other 2 things, is that DNA is transcribed into RNA, which is translated into protein. And that's again happening, this is where protein's being made. It's happening at the ribosomes. And a shorthand way of saying this is the DNA is being transcribed into RNA, which is being transcribed into proteins. And hopefully you remember that as the central dogma in biology. But that's all happening in G1. It's a very busy phase. It takes up a lot of time in a cell's cycle and its cell's life. In the S phase, I'll do this in a slightly different color. In the S phase, this is where DNA is copied, right? So cells are making an extra copy of their DNA, which I hope you recognize, or maybe you're asking yourself, why is that important? And I hope you recognize then that it's important when the cell divides that it has an extra set of DNA to give away. And that's what's happening here. So DNA is being copied. The name of that process, which we will talk about soon enough, is called DNA replication. All right, so this is where DNA gets copied. And G2 is, there's a lot going on in this whole thing, but let's just simply know this is when the DNA copies are proofread, right? So they're making sure that they didn't make any mistakes. Although I should point out that sometimes mistakes happen, and if they happen and they get passed down, we call those things mutations, right? But cells are usually pretty good at catching them, although they're not foolproof. All right, so this DNA then is going to be copied. But before we jump into this last quarter of the cell cycle, I will tell you that if for mitosis, let me remind you, this happened in eukaryotic cells, right? In eukaryotic cells, and this is where we're going to dive into something that's specific to those eukaryotic cells. So what I'm going to do is pick a color, pick this green color, for mitosis. Let me pick the same color that I used the title in. Mitosis, and what I'm going to do is do another dotted line, and we're going to divide that into 4 parts. So again, we're going around clockwise. So the first stage, all of this, let's just say all of this is mitosis. Notice that I did leave a little sliver out. We'll talk about that. And the first part of that first is a phase called prophase. Then we have the second part is metaphase. The third part is anaphase. And the fourth stage is telophase. But this last section, which is not included, in this case, it's not included in mitosis. We'll talk about that in a moment. Is called cytokinesis. Right? And cytokinesis, if you were to break down these words, kinesis or kinetic means movement, cyto means cell. This is actually where we describe the cell breaking apart. All right, the cell breaking apart. Whereas this mitosis, all of this stuff, mitosis is really about nuclear division, right? Nuclear division. So this is where the nucleus divides. And I think that's critically important for you to understand, because sometimes, actually more often than not, or no, I take that back, more often than you probably think, mitosis happens and it skips over cytokinesis altogether. And we actually end up with, if you think about this, if mitosis happens, we have a nucleus that divides into 2, and then the cell doesn't split, so the cell then has 2 nuclei, and we describe that kind of cell as a multinucleated cell. And I want you to ask yourself, what kind of cells in your body would have multiple nuclei? What function would that serve? How would that be beneficial to some types of cells? Okay, and if you can't think of that, ask me in class. Excuse me. So what I want to do is actually dive into what these cells look like, so you can actually tell the story of what's happening here. And there are a couple of key words that we need to discuss. So we need to remember we got prophase, metaphase, anaphase, telophase. You will need to remember that. And a good way to remember that is just, well, a good step in the right direction would be the first letter. And you might think of that as PMAT. Prophase, metaphase, anaphase, telophase. There might be other ways to remember it, but remembering PMAT alone is not sufficient. You got to remember the name of it and be able to describe what's happening. So let's go ahead and start making some drawings of what's happening in each of these stages of mitosis. So let's remember that we've got prophase, metaphase, anaphase, and telophase. Right? And all of this, sometimes telophase gets combined with that cytokinesis, but it doesn't have to happen, and that's why I like to separate it. But in prophase, we would start with a cell. Let's go and label this as prophase. We start with a cell, and we'd find all these organelles, the normal eukaryotic organelles in there, but I'm not going to draw them all in here. In fact, I'm going to make that a little bit bigger, because I do not like, I want more space. So if I have a cell that's here in prophase, I would have a nucleus. I would have, let's go and draw that nucleus. I'm going to draw it as a dotted line. and I'll explain why in a second. And inside that nucleus, I'm going to find some chromosomes. Remember, this is a eukaryotic cell, and that DNA actually creates what we call chromosomes, which I think you know are drawn like this, right? And so I'm going to draw this cell with 4 chromosomes. And these 4 chromosomes would represent the amount of chromosomes in each cell. So this organism, this cell that I've just drawn, has 4 chromosomes. If we were to think about a human cell, I hope you would know already, but if you don't, we would expect to see 46 chromosomes. I hope you understand why I don't want to draw 46 chromosomes here, because it's going to get a little bit more complicated. What's happening here in prophase, though, is the cell's getting ready. These chromosomes are actually forming. Before this point, the DNA actually exists in a form called chromatin. So let me go back here. So in all of interphase, DNA is in the form of chromatin, Tin. And the way to think about that is that DNA is kind of loose, it's open, it's unwound, and that's critical because in order to read the DNA into RNA and then into protein, it has to be open, has to be available to be read. That code has to be read. Also, if DNA is going to be copied, it needs to read every section of that code and it needs to be available to be read. But what happens here, right about here in the cell cycle, DNA condenses. All right? DNA condenses up into chromosomes, which is what you might have learned about in middle school. And those chromosomes look like those X-shaped things often. And so this is what we might see inside of the cell. The nuclear membrane in prophase is starting to break down. And what we'll also start to see on the edges, maybe not on the edges quite yet, but that's easy enough for us to work around. We end up seeing these things called centrioles. And centrioles are these organelles we haven't really talked about that will end up shooting these proteins out called spindle fibers that grab the chromosomes. And so I think it's going to be really important for us to really think about and understand the anatomy of this chromosome. So I'm going to draw one down here. All right, so I'm going to draw these 2 sides of this X, and I'm not actually going to let them touch, although we might want to think about, oftentimes there's this bump, right, and that might get colored in, that attaches these. And so this middle part is called the centrosome, or the centromere, right? Central, right? It's a central section of this chromosome. Alternatively, the end of it you might have heard of, especially if you kind of have a better understanding of aging cells and cancer, is that these ends are called telomeres or telomeres. But the 2 sides of the chromosome, that's what I really want to focus on right now. These 2 sides, this yellow strand on the left and the yellow strand on the right, are identical. Right? They're identical, aside from some mutation that might have happened. They are identical. And they were actually made during the S phase of interphase. So this is the copy of the DNA that was made. Right? So that they were copied, and we have a special name for them. And they're called sister chromatids. Right? Sister chromatids with a D. And together, when we have 2 of them, we call this thing a replicated chromosome. So that's going to be important for you to conceptually understand because we're going to rename chromosomes in a few minutes or redefine them. And so a replicated chromosome has the two replicas or the two copies of the same chromosome. What's happening in prophase 1 is the membrane, the phospholipid bilayer of the nuclear membrane is breaking apart, getting ready for metaphase, right? So metaphase, we have this cell. It's going to go from prophase to metaphase, and the nuclear membrane is gone, and we actually end up seeing all of the chromosomes lined up in the middle because we have these new things called spindle fibers that have shot out, these proteins, these filaments of protein that have shot out of the centromere. They grab onto the centromere of the chromosomes and line them up in the middle. So we have 4 in the middle here. In a human cell, we'd see 46 lined up. Although they look like a single file line here, don't forget that cells are 3-dimensional, so we would actually see them all on the same plane, right? We would see some behind the others in a 3-dimensional representation. All right, so we have them lined up in the middle, and then we move on to anaphase. And in anaphase, we see them actually start. We see the sister chromatids. Oh, that's bad. Okay. Actually come apart. Right? So what we'll actually see is that these centrioles will have used their spindle fibers to pull apart the chromosomes, right? To pull apart the sister chromatids. Now, don't worry, because each of these 4 chromosomes that each had different genes and different codes on them, they had replicas. And now this side of the cell has the exact same code as this side of the cell because the replicas have been pulled apart. Which then leads us into this next stage of telophase. And in telophase, what we would see is the nuclear membrane starting to reappear. And inside of the nuclear membrane, we would see the 4 unreplicated chromosomes, right? So I think I want to take a moment and have you count how many are in each of these 2 nuclei. You might say, wait a minute, there were 4 to begin with over here, 4 X's, and now I only see 4 halves of X's. But I want you to remember that this had 4 unique sets of information, and here we still have those same 4 sets of unique information. So this right here, so I want you to know that this is an unreplicated chromosomes, but they're still chromosomes. So there are 4 chromosomes at the beginning of prophase, and there are 4 chromosomes in each of the nuclei at the end of telophase. So we did not change our chromosome number. I know that's a little weird and confusing, but remember that this cell is going to continue through the cell cycle. So after telophase, this is the end of mitosis. But what sometimes happens, not all the time, what sometimes happens is that cell will split apart. And so we actually end up with a nucleus over here, maybe a nucleus over here, and inside of each of those nuclei we see 4 unreplicated chromosomes. And what you might imagine is that there is this little bit of a bump in right here. Right, so I'm going to use a better color. So we call this thing a cleavage furrow. And what we'll see is that it'll keep pinching in, Eventually, closing off, and we end up with two actually identical cells. Same code over here, same code over here. And sometimes telophase and this, which is called cytokinesis, happen at the exact same time. But remember, cytokinesis is about the cell pulling or tearing apart, and telophase is the two nuclei appearing. So they're two separate processes. But this cleavage furrow is only true for animal cells. I will point out that for plant cells, we would see actually, when we typically draw plant cells as more more rectangular, we would actually see what we call a cell plate form, and it's made out of, it'll be eventually form into like a cell wall, and it's got lots and lots of cellulose. So basically what we would be seeing is lots of sugar being built up on this cell plate, dividing these two cells with two different nuclei, each also with the exact same chromosomes and same code. Excuse me. But after this, it's important to know what the cell is going to do. We've got these two smaller cells that are then going to go back through the rest of the cell cycle. Each of those cells are now going to go through G1, where those chromosomes are going to unwind. So we end up back with DNA that's in the form of chromatin, so the DNA can be read into RNA into proteins, and then it's going to be copied again. So that later when it condenses back up right before mitosis, we end up with both sides of the X, and we have a replicated chromosome. Okay? So the big takeaway here is that in order to divide these chromosomes, we have to copy them, right? And then we divide them, and we copy them, and divide them, and copy them, and divide them. And that has happened trillions of times to make your cells today, which is one really cool idea, maybe unsettling, is that the original cell that made you up is no longer around. It's died. It's long gone. But the code that was in it, that was made from dad's sperm, mom's egg, is still replicated and copied and split in you, right? Those new cells that your body forms carry the same code that was part of your origin, part of your conception, despite the fact that we can't find that original cell anywhere in your body. I think that's kind of a neat idea, that it's really just about the passage of DNA. Now, what's really kind of neat is that we can look at the proportion of cells in some tissue, some part of an organism, and find out how quickly it's growing. So normally a cell would spend about 90% of its life in interphase and a short blip of time in mitosis and cytokinesis. But if we were to look at a bunch of cells and we saw a bunch of them in mitosis, that might mean, based on some idea of probability, that that organism is growing really quickly. So we could look at root tissue, we could look at leaf tissue, and we could see in your crops if there's a higher, what we call a mitotic index, right? So I'm going to add that. I'm going to scroll down just a bit here. I'm going to add this into my notes down here, that the mitotic, that the mitotic index is a measure of the number of cells in mitosis compared to the total number of cells, usually times 100. So you look at what percent of the cells are in mitosis. That's the question we're asking. So the more cells that are in mitosis, the more quickly cells are growing. I hope that makes sense. And we can look at that through the microscope and identify cells that are in any of these stages where we can see chromosomes. If I can see chromosomes in an X shape, it's in mitosis. And I can count up how many cells I see that show me those chromosomes versus the cells that I can't quite see that in, or it's a little unclear. So in interphase, let me just tell you real quickly that we would see a cell in interphase would look, we've got this membrane, we've got a nucleus, and we'd see a dark region inside that nucleus. And that would be it, right? Or maybe the nucleus, the whole nucleus would be dark. But either way, we can't see those X-shaped chromosomes because the DNA hasn't condensed into these large bundles of DNA. And so it's so thin that our naked eye, or even with the microscope, we still can't see it. All right, so I'm going to stop there. I'm at 30 minutes, but I hope this is useful and a lot of information, and I hope you have a lot of questions.